Hello everybody, my name is Dane and welcome to this event about what's inside us and how is it made. It's a really nice opportunity to be working with Shankar and the Shivanas group and Tomoko on some of the really interesting research that they're putting together and just explaining in really beautiful ways. And this talk will share some of that. So the format's going to be Shankar talking about some of the work at the Department of Physiology, Anatomy and Genetics, and that will be 20 minutes, half an hour, and then we'll have some questions, so please get them ready. Enough of me though, I think it's time to crack on, and it's a real pleasure to welcome once again Shankar to the IF Oxford stage. So Shankar, over to you.
Thank you very much, Dane. And thank you to everyone in the audience for coming and joining us today. Very briefly, in terms of an introduction about myself, I'm the professor of developmental biology in the University of Oxford, and so my group in the university works on how embryos develop. In addition to that, I'm the medical fellow in Jesus College, so I teach the medical students aspects of embryology during their first year, and also aspects of embryology in their third year. So today I'll be telling us about the lives of the cells within us, and also actually alluding when I have in the title a look at the life inside us. I also speak about during pregnancy, the new life that women have the privilege of carrying within them to term. And we study how the embryo, the baby, develops during this process, the very earliest stages. So I'll tell you a little bit about that aspect of our work. So to begin with, the body is made up of cells. And this slide is meant really just to illustrate the diversity of cells in the body. It shows forming a tadpole, so a frog embryo, and you have what's called a zygote. The zygote is the name given to the fertilized egg. And the specific names that they don't really matter. It goes through a variety of embryonic stages. And what this diagram is really meant to illustrate is that, of course, we're not just a sack of identical cells. Cells specialize so that they can perform their functions properly. Or optimally. So you have heart or muscle cells which are specialized for contraction and generating force. And you have, say, red blood cells, which are specialized for carrying oxygen and CO2 around the body. You have neurons, which are specialized for transferring information, electrical signals, essentially, from one part of the body to the other. So these are all specialized for various tasks. And so that's one of the earliest things that occurs within your body during development. Development is the process by which you go from the fertilized egg to the final fetal form. And indeed, development obviously continues after you're born as you develop as a child into adulthood. So one of the first things that jobs of an embryo is to divide, make a diversity of cells. But as I said, we're not a sack of cells just mixed up at random throughout the body. We are exquisitely patterned. We have shapes, we have different structures, we have different organs, which are collections of cells of specific types. So in the olden days, they used to think that there was a tiny homunculus inside each and every sperm, and that during embryonic development, this homunculus, this preformed little person, grew in size. And so, of course, all of the information lay in the male cell, the sperm, and the ovum, the female cell, the egg, was responsible merely, so to speak, for providing nutrition and sustaining the growth of this preformed human. We of course know that there's nothing further from the truth. Somehow you go from an amorphous, largely spherical egg, fertilized egg, into a finely patterned organism, a finely patterned fetus. And when I say pattern, I mean in terms of shapes and the arrangement of cells. This then further goes on to give rise to the newborn, and then later develops into an adult. Just to bring this home to you, the example I always give is of the boiled egg you may have had for breakfast this morning. It looks, of course, nothing like a chicken or a chick. But somehow, reliably and reproducibly, if given the right temperature, that's all you really need, a chicken egg will transform into a chick. Almost seems like a magic, but ultimately it's a chemical reaction. And that's what we're trying to study, this series of steps, reactions that occur time after time after time, given the right conditions, to reliably give you the final form of the organism. One other point I always make, though we take it for granted, but we perhaps take it too much for granted, so we don't sometimes recognize it, is that a chicken egg will only give you a chick. It'll never give you a puppy or a kitten. So again, this points to how this is essentially programmed into the egg, into the material of the egg, so that it reliably gives you one thing, the chick, and not something else. It's not something that occurs by random chance, and therefore it is tractable to study, which is what we're trying to do, to understand how this comes to be. So in this movie of a tadpole embryo, I'm just showing you on left, you can see the cell, the initial egg dividing repeatedly. And as the cells divide, they're not growing in size. So each individual cell becomes smaller and smaller and smaller. And this is just to illustrate one of the earliest events, as I described, to cell division, so that you can generate the material with which to form the embryo. But I said the cell movements are responsible for shaping the embryo. So it's not as though cells are already preformed, or sorry, the form of your body, the shape of your body is already preformed. That's not the case. You need to have coordinated movement of these cells so that you have the various organs in their correct locations, and indeed so that cells are separated, so that muscle cells are only in your muscles and not in your brain, and vice versa. Neurons are only in your brain and other parts of your nervous system, and they are not in your liver, for example. So this type of movement is illustrated in the movie at right, showing now again a tadpole. This movie starts where the embryo already has many cells, and you can see the types of cell movements that go to shape this early embryo into, now you can just about start to see at the end of the movie the beginnings of the shape of a tadpole. Here's another example of the type of cell movement that go to shape, in this case, an organ, the kidney. And this is a movie made by Tomoko Watanabe in Frank Costantini's group quite some time ago. Tomoko is works in my group now, and she's one of the main people behind setting up our stall. And she will be available at various points for you to discuss with her, if you wish, or me, our aspects of our work. But so the movie at left shows you how the kidney forms. And the kidney forms by a process that's described as branching morphogenesis. In many ways similar to the way branches of a tree, they branch and give you the final shape of a tree. And depending on the way those branches occur, you'll either get the typical shape of, say, a pine tree or an oak tree, completely different shapes. And that's determined by the pattern of branching. And you can see how these cells, it's very, very fluid. This branching is what sets up the early kidney-shaped, the kidney-like shape of the developing kidney. So how do we know all of these things? How, in fact, here's this movie. How did we make this movie? And this is all about comes back to the title of the talk, watching, seeing what's inside us. So I'll tell you a little bit about some of the technologies we use. And these are, of course, very tiny structures. So we use a microscope. In front of you, you can see a standard microscope. You can see the two eyepieces through which you could look to see things by yourself if you need it. But you also have to record movies. We use digital cameras, very similar to the cameras you have on your iPhone or a smartphone, except just much more sensitive and therefore sometimes bigger. So that's in this big black box here. And then we need to culture these embryos in the right conditions. Especially if you're studying, say, mammalian embryos, which are accustomed to developing inside the womb at body temperature, roughly 37 degrees, depending on the animal concerned. So this plexiglass box here helps maintain the correct temperature around the entire microscope. And you would put your sample over here on the stage, and I'll show you in the next slide, which you can zoom in. That you put your sample here on this stage, and you can allow the sample to culture and develop. You can take embryos, even those that are accustomed to growing inside the uterus as in mammals, and you can culture them on a microscope for limited periods of time and observe their development. And this is just showing you also that you can supply various gas mixtures, which are again important to enable them to develop normally. Another important aspect that has facilitated technology that's facilitated us facilitated the visualization of development and the cell biology is green fluorescent protein, or GFP. This is first isolated from the jellyfish. And in fact, it won the scientists Osamu Shimomura, Martin Chalfie, and Roger Chen the Nobel Prize in 2008. Basically, GFP is a protein, and the special property of this protein is that if you shine light on it of one color, it will emit light of another color. So if you shine blue light on this protein, it will emit green colored light. Hence the name green fluorescent protein. And so jellyfish, you might be familiar with bioluminescence. Many aquatic life forms can glow either, so it can be really a magical experience sometimes if you had the pleasure, the luck to have experienced it, where you just move your arms through the water and it you can set off this train of glowing sparks. Those are tiny organisms that are putting out light. So GFP is designed as a protein to do this, and it enables many of these the jellyfish in this case to either signal a warning or its presence to other potential mates, etc. The reason these three gentlemen won the Nobel Prize is they realized the potential of this protein for experimental purposes. It so turns out that you can take this protein and you can actually put it into other animals or other plants. In fact, you can see here at the left is a plant called Arabidopsis, which is related to the mustard, and it's used a lot in research. At the top left here, you can see various bacteria. At the top right is a developing zebrafish, and at the bottom right is a mouse. So you can take the gene that codes for the green fluorescent protein, and you can make genetically modified organisms, in which all of these examples are, to put this jellyfish gene into these animals or other plants or bacteria, and they will make the protein, and they will have this protein, again, basic chemistry. It behaves this way in jellyfish. It'll behave this way in a mouse or a zebrafish or a plant, such that if you shine light of one color on it, it will emit light of another color. This is useful in the context of microscopy because it then allows you, if you were just relying on light passing through your embryo without any of these labels, you wouldn't have contrast. In fact, often more than brightness or magnification, you need contrast. Contrast is what allows you to discern one region from another region. And contrast allows you to then see details. You can use this green fluorescent protein and make sure that it is present or it is functional only in a subset of the cells of the sample. And that allows you to see that sample in better contrast under a microscope than you might otherwise be able to. So you can see here, for example, in the case of the zebrafish, the green fluorescent protein is mostly in the forming embryo and not so much in the yolk, which is this dimmer structure here. And you can see in this mouse that most of the GFP is expressed in the ears and in the snout and in the paws and less in other places. So it allows you to then essentially, it really enables you to visualize things much more in detail. One more thing about GFP is that since its discovery, there have been many variants of green fluorescent protein that have been engineered in laboratories across the world. And these give you different colors. And so you can see here small tubes with solutions of variations of GFP showing you different colors. And this allows you, therefore, then to be able to see multiple structures within a single sample so that you could label one structure in green, another structure in red, and see how they interact or how they move in a coordinated manner or not coordinated during the development of your embryo of interest. And then finally, in terms of the technology, I just want to briefly mention other types of microscopes. I showed you a microscope which is recognizably a microscope, the image I showed you earlier, where you had eyepieces which you would look down to see your sample. But many modern microscopes, they dispense with the eyepieces altogether because there's, though it can be helpful to be able to see something with your eyes, in modern research, you need to actually record the development of your sample, and you can use very high resolution cameras to be able to record, and very sensitive cameras to be able to record your sample. So you don't really need an eyepiece because ultimately you're going to be digitally recording the image or the movie, and then coming back and viewing that movie. So here's a microscope at left. It's a type of microscope made by Zeiss called a light sheet microscope, and this is what it looks like. It's a box. You'd open this gray lid and put your sample inside. These blue structures here and here, those are the digital cameras that allow you to capture the image, and the whole thing is computer controlled. It's all completely fly-by-wire. You control everything from your screen. These type of microscopes allow you to make really very detailed movies. This is a movie of a zebrafish embryo developing, and each of these blobs is a single cell. This is, in fact, the nucleus of a cell. The nucleus is what contains the genetic material within the cell. And you can see every single cell that makes up the body of the developing zebrafish. You can see the movement, and you can track them in great detail as they form the zebrafish. And you can see, hopefully, the structure, the features of the developing structure. Of the developing, embryo. This is the developing body of the embryo, and the rest of it is the yolk. And this, the animal view and the vegetal view at the bottom, that refers to two different angles with which you can view it. This type of microscope, it has an advantage. That's why it has two cameras. You can actually simultaneously view your sample from two different angles, which gives you, of course, more detail about the whole embryo. So I'm going to stop now with the general background and move on to tell you a little bit about the specific type of the questions that my group works on using some of these technologies that I just described. So the two main questions that we address is how the basic body plan is laid down. And by basic body plan, I'm referring to what is called the rostral caudal or anterior posterior axis, the head-tail axis. Naturally, the body is different from the head to the tail. So, and that those differences are amongst the first to be laid down during embryonic development. So we work on how that axis is first formed. And the other main question that my group works on is how the heart forms and starts to beat. So how is it shaped? and what triggers, what sparks the earliest contractile activity of the heart. And at left here, these are both mouse embryos, and at left you can see a movie of a very early mouse embryo. And these cells in green, you can see them moving. That movement is essential in setting up this basic body plan, the head to tail axis. And the movie on the right is actually a developing heart, and I'll tell you more details about it as we go along later. But to begin with, again, as an overview, so in my group, to address those two questions, we use the mouse and human embryos as model systems. Most of our work is on the mouse, but we do a little bit of human embryo work as well. And you can see here in the middle a very early human embryo, roughly day 17 to 20 days after fertilization. And here at right is an image of twinned mouse embryos, where you can see that, of course, in both of these cases, both the human and the embryo and the mouse embryo, they look nothing like, of course, the form of shape of a fetus or a baby or an adult human. And that again gets back to my point that the cell movements during embryogenesis, they're crucial in giving it the final shape of your body, which is what we study. So this example of mouse embryos, they're actually twinned, so they're actually connected here at the forming placenta. But this would form one mouse embryo, and this would form another mouse fetus. At left, I just have a diagram of the life cycle of a mouse from fertilization to adult. And this to illustrate basically that the mouse mouse development is a suitable model of human development, though it may not appear to be at first glance. From fertilization to birth in the mouse takes on the order of 18 to 20 days, depending on the strain of mouse. So though that doesn't in any way compare with the 9 months of human gestation, at early stages, there's much more of a link between the staging and development. Implantation, the process by which the fertilized egg implants in the lining of the womb, the uterine lining, that takes place. The implantation takes place around 4 days after fertilization, between 4 and 5 days in the mouse. In the human, it takes place around 6 days, 5 to 6 days after fertilization. So not that different at that early stages. There's a key event during embryonic development called gastrulation. I won't go into the details of this event, but suffice it to say, it's the event during which many different cell types are laid down, and much of the early shapes are laid down as well. That starts around 6.5 days after fertilization in the mouse. In the human, it's around 2 weeks after fertilization. At the early stages, the process that of morphogenesis is very similar between the mouse and the human. And in fact, unfortunately, I don't have them with me here, and even if I did, it would be less useful. But normally, when we have a stall at the festival, at the science festival, we have these 3D printed models of a mouse and a human embryo. At the equivalent stage of development, and most people guess, when we don't tell them which is the mouse, which is the human, and ask them to guess, most of them get it wrong. And as often as not, they think that the mouse embryo is a human, and the human embryo is the 3D printed human embryo is the mouse. So they look very similar at these early stages. So we use a mouse as a model, as one of the main models. And now I'll tell you two brief stories about some of the research in the lab. And I'll obviously be available to take any questions, specific questions you have about them. So I'll go over them very briefly to leave time for questions. Before that, science is a very collaborative endeavor. It's not at all, has perhaps a stereotype of a scientist being socially inept, which many people perhaps are, perhaps not. But I don't think any more so than in other areas of life. Or of science being a solitary undertaking, where a person working in a laboratory late at night all alone. There can be nothing further from the truth. People do work late at night and all times of day and night, but the work is very much collaborative. And the work I'm going to be telling you about today involves the two stories I'll be telling you about. And one is a collaboration between Matthew, Felix, and Holly with Jason. Matthew is a biologist, and in fact, the other the three, Felix, Holly, and Jason, are all computational people. And I'll tell you about this wonderful collaboration between them to try to understand cell movements. And the second story I'll tell you about is the work of Richard Tizer as well, working with the members of my group and collaborators elsewhere as well. So in terms of setting up the head-tail axis, how does that form? The mouse embryo, very early on in development, looks like a cylinder. Okay, it doesn't look anything like a mouse. But here's a movie of this type of a structure. This is one of the earliest movies that I ever made. And it is the first movie of this process, whereby these green cells, I won't go into the detail of their name, but these green cells move from one end of the cylinder up to one side to midway along the length of this cylinder. And this movement is essential for the specific orientation of the head-tail axis. There are various genetic mutants where this movement fails to occur, and these embryos fail to progress further in development. Okay, so this movement is absolutely essential. So we're studying how this movement occurs, both to understand the process of the head-tail axis formation, as well as to understand how cells move during embryogenesis. And to do this, we've been imaging embryos. And here's a more detailed set of movies using a light sheet microscope, which I showed you a moment ago, where you can visualize the movement of the individual cells during the process of the migration. And again, these cells express GFP, so that you can see them under the microscope very clearly. You can see the outlines of the individual cells in this case. And you can also tilt you capture essentially the entire three-dimensional volume image of the embryo, and then computationally on a computer you can tilt it and look at different angles. You can zoom in on it and watch cells divide. As you can see over here, you'll see two cell division events where these two big cells very rapidly, you can see them balling up now and ping, dividing, and now here's the second cell division. So you can visualize cell divisions, you can get a great deal of detail. But we want to be able to then now analyze this type of behavior in a quantitative way. We want to measure things, see how fast they move, the path that they take, etc. You can do this manually. You can put dots inside individual cells and manually see how they move. If you're interested in cell shapes and cell divisions, you can outline cells manually again, and you can score dividing cells, etc. If you want to look at the volume of a cell, you can actually take the volume, the stack of images that make up your volume, and you can outline them in 3D, and you can measure their volume, etc. But doing this manually is tremendously time consuming, so much so that it makes it completely unfeasible to study this in any way. And as an example, if you had the 200 cells in the embryo that you're imaging every 5 minutes over a 10-hour period, you need to essentially sit, a human would have to sit there and outline 24,000 different cell outlines to be able to manually look at each of the individual cells. So this is not really feasible. So we try to automate this. And to automate it, we use various approaches. One approach just shown here is an optical flow approach developed by Felix Su, very published in 2019, where you can use computational approaches to look at the movement of cells. And you can see here now the group of cells that I originally showed you, the green cells which specify where your head will form. We can now look at their movement and with respect to the movement of the surrounding cells, and it tells us basically that it's only they're so-called AVE, anterior visceral endoderm, that it's only those cells that move and not the surrounding cells. We also try to analyze this in flat projections. As I described, the embryo at the stage is a cylindrical structure, and it can be difficult to visualize cell movements in, if you're trying to look at both the front and the back of the sample at the same time, it can become really difficult. So what we've also done is use existing mature mathematics to unpeel the surface of the sample. So this is exactly what's done for when you make maps of the globe. So the globe or the Earth, of course, is spherical. Excuse me, I'm just going to have a drink of water. Excuse me. So the globe, of course, the Earth, of course, is a globe, but the maps are flat. And there are various ways of taking the surface of the Earth, of the spherical Earth, and projecting them. In this case here, a flat projection. Here's a polar projection, etc. So you have Mercator, Cartesian, a variety of different ways of projecting these surfaces. So we did exactly the same thing with the cylindrical embryo, either as a flat rectangular surface projection or as a circular polar projection to look at the tip. This curved tip of the mouse embryo. This is the type of result you get. This is one face of the cylindrical embryo. And here, in this case here, the surface has been unpeeled and laid flat. And so this will be the front and the back of the embryo. And of course, this portion here where you cut the view comes back and joins at this point here. Okay, so there's a continuity. So it makes circle and around. So you can now again see all the cells. And here is a polar projection looking at the tip, the curved tip of the cylinder, and you can visualize some movements like this. Now, to be able to automate the outlining of cells, we actually need better quality images than we are able to capture. When capturing these images, we always need to make a balance between the quality of the image and the survival of the sample. Because when you shine bright light on your sample, they actually warm up. Just like even a light in the room, if you put your hand close to the bulb, you'll feel the warmth of the light. So when you shine light and look at embryos, they warm up and they don't like it. And they don't develop well. So we keep our light low, which means that the quality of the image is not as good as it could be for automated outlining. And so we use machine learning to actually clean up the image. Similar to, for example, the algorithms that Google or your phone photograph album might use to recognize faces in a photograph. You can train the computer to recognize the outline of cells. And then from a grainy image like this, it can transform that and give you a good signal and high contrast, so you can see the outlines of cells. And this enables us to then make these type of detailed movies of the movement of cells and visualize this movement in good detail. This then allows us to really start analyzing how these cells move in a coordinated manner and start to discover the we get new insights into the coordinated movement of the cells by plotting in quantitative detail how the cells move. Okay, so I just wanted to illustrate from this small story how there's actually the biology we do is very interdisciplinary, as much of modern biology is, and involves collaboration between biologists, microscopists, engineers, computational scientists coming together to make these advances. I'll end with a very brief description of the type of approaches we take to study how the heart starts to beat. And in this movie, you can see an early heart forming, which I'll show you in more detail in the next slide. In this, basically, you're viewing a mouse embryo frontally. It's semi-transparent. These are all the same movies showing different components of the embryo. In the green part here is the outermost layer of the embryo, and here is just an overlay of the green on top of what is called the black and white image, the bright field image. In this embryo, which is movie which is looping, this dark region here, that's will form the heart, and these ridges over here, those will form the brain. And I just want to illustrate that during embryonic development, this is real and true, and this is true of mice and humans and most mammals, that your heart actually starts out ahead of your forming brain. And it's these type of complicated movements that we study that bring it to its lower position in the fetus and the adult. But what I'm going to tell you is not about how the heart forms. For the purpose of brevity, I'll tell you a little bit about our work on studying how the heart beats and when it starts to beat. So we were able to culture embryos at various, over the course of early development. And so this is a single embryo, and each of these is a movie at different stages in culture of this particular embryo. And you can see here, this tells you how long it's been in culture. And you can see that this is the forming heart. Again, it doesn't look like anything like a heart, but again, the same way the embryo doesn't look anything like a fetus, this will go on after a lot of changes, twisting and turning to form the more recognizable heart shape. But you can see that it's already started to beat. And if you go back in time, you can see that at this very early stage where it's what's called a cardiac crescent stage, just the faintest twitches over here where my pointer is, you can see that already at this early stage, the heart, the forming heart beats. Just going on to the next slide. It's known, it's very well established that calcium ions, calcium Ca2 plus, is important for muscle contraction, whether it is your skeletal muscles, the muscles of your arms, for example, or whether it's the muscles of your heart, the cardiomyocytes, and the cardiac muscles. So calcium is known to be essential. So we also used, again, light microscopy and fluorescence microscopy to be able to see calcium within the cell, to look at the levels of calcium within the cell. And in fact, it's in a periodic manner, changing the amount of calcium in a cell very quickly on the order of seconds by bringing the level of calcium up, bringing it down, bringing it up, bringing it down. You can actually make the muscle beat in a periodic manner. And looking at the early embryo in this way, using a dye which allows us to see the levels of calcium in the cell, we were able to see that you have these synchronized waves of calcium already at these really early stages during embryonic development. But interestingly, before you have these synchronized waves, you have these asynchronous, random oscillations of calcium within individual cells. So one thing which we're now studying in the group is to see how these individual random oscillations of calcium get synchronized during the course of embryonic of heart formation, so that you can get a nice synchronous wave of calcium, which leads to a nice synchronous wave of contraction across the forming heart. And the reason this is really important is that if your contraction is not synchronous, the heart will not be able to beat properly. You have things called arrhythmia, which can cause your heart to stop beating altogether. So it can be really very bad if it happens in the adult. And so it's interesting to see in an embryonic context when how this rhythmic contraction is first established, and hopefully by understanding how this rhythmic contraction is first established, we will get a little bit of an understanding of how it may go wrong in adults who may suffer from arrhythmia. So I will stop there with just by ending by showing, acknowledging some of the people who contributed. As I went, I already described the main people who were part of this work. Sorry, as I went along, but sorry, but yeah, but here is just a photo of our entire group during a Christmas lunch last year, and a number of collaborators based both in Oxford as well as in other places around the world. And various of the funding bodies who enable this work to take place. So I'll stop here so that if you have any questions, I can try addressing them. Thank you.
Thanks, Shankar, for that. That was a really fascinating and very rapid tour of the developing mammalian body. Tomoko has been really helpfully answering the Q&As live, but there was a question in there that has been answered that we'd just like to kind of throw your way. So there was a question about which came first, the sperm or the egg, the chicken or the egg.
Yes. I'll answer that in a literal sense. Of course, neither of them, you can't speak in terms of either of them coming first. You look, there's a continuous progression of life across all the related living beings on Earth. And so before the chicken or the chicken egg would have been the last thing that you might have considered to be a relative of the chicken, but not quite a chicken yet. Which you would have imagined would have laid the very the one egg that then founded all chickens to come evermore after that. So arguably, you might then say it is the egg that came first, because of course there would have been an egg that would have been laid by something that was not, that you would not consider to be a chicken, and that egg would have given you a chicken. So yes, I mean, I guess, being forced to answer that, if I had to choose one or the other, I would say it is the egg that came first, not the chicken. But of course, you can see it's a continuous process. We're all related across time, and so there's a this concept of the most recent ancestor, the most common ancestor. So if you just go back in time, everybody is related. Everything is related.
I think that's a really good answer. So I'm going to now write a press release saying University of Oxford professor says it came the chicken, the egg was. But I suppose the thing that was first just about more noticeably than an egg than the previous thing that species produced to generate offspring, we've now decided we've just had an event about the planet Pluto being demoted from being a planet. So this kind of fine gradation of, the sort of technicality of, was it an egg? Was it a chicken? Was it not an egg? So thank you for that explanation. So some of the other questions we've had was around what does a body look like before it's born? So you mentioned the kind of homunculus was this kind of old-fashioned idea of a tiny adult, with a briefcase and a haircut, kind of coming out of the womb. So at what point would you say? I mean, obviously you've shown it in some of the slides, but at what point would you say this thing is now very, very recognizably a human sort of shape? Like in terms of the gestation period in a womb.
Yeah. No, when you say in terms of recognizably, it depends. I mean, for example, the human embryo has a tail, but which is lost during embryonic development. But already easily by say 2 months, in the second month, you'd have something that looks, it has a head end and a tail end, recognizably something that looks like a head end and something that looks recognizably like a tail end. You could imagine that by around 2 months, 3 months, it's curled up in a fetal position. So you would be able to make a guess. Even what I mean to say is, even a person who is not familiar with what an embryo looks like would probably be able to make a guess at what is going to be the back and what is going to be the front. But it looks nothing like a human. In fact, I mean, I would actually argue and say, when you say, what does the human body look like before it's born? Obviously, the baby doesn't look anything like an adult. Here's something, again, some people may internalize this. Certainly people who have children or have recently had newborns, this might be something which you're easily able to relate to, but many people will not. And even if you've had children, you may not be able to. A baby will not be able to touch the top of its head. Simply because the arms are not as long with respect to the size of the body as they are later in development. And I don't know if you can see me, but you can see that I certainly, and I think most adults will be able to touch the top of their heads because our arms are long. So the body grows at different parts of the body grow at different rates. Again, if you look at a baby, the head is very big with respect to the rest of the body. but as you grow, the head doesn't grow quite as much as the body. So even the human body changes after birth, and it's continuously changing during before birth and after birth.
Okay. I think even in some countries when it's, or some places or periods of time when things like birth certificates haven't been sort of documented, isn't a way of kind of working out the age of a child is whether or not they can reach their shoulder or something, or do that kind of touching the head business. Is it sort of, how old is that, sort of 6 or 7 or something like that, when it's just about enough, when the relative length of the arm is that?
I don't know the specific thing. Again, it'll vary from, of course, it'll vary from child to child, but probably by around then they are able to touch the top and perhaps the shoulder, the opposite shoulder.
Yeah. I think that's just also looking at the really rather beautiful kind of images of kind of growth and how things don't just develop in the womb and how people obviously change during a life course. But I was really taken by I think it was some of the imagery that Tomoko kind of made that you showed of was it the liver, the sort of branch structure kind of developing, and sort of how these sort of shapes grow. So maybe I don't know if Tomoko is there, if she could maybe just talk about how did you make that video?
So embryonic kidney is one of the few organs that you can grow in vitro. So you can dissect out a embryonic kidney, like when it's starting to bud out from a tube, and then you can just grow it on a filter under microscope and just observe it over time. So we have the GFP growing inside the ureteric bud, which is the tubular structure of branching, and that's how we could see the white, the grayscale, the white bit, but it's actually encased in a mesenchyme tissue. It's a bunch of ball of tissue that interacts with the branching structure to encourage the growth and then the differentiation of each other.
It was again relating to the point about the contrast I was describing. So in fact, though you would only really see this branches, which the way I imagine it almost looks like a toothpaste being squeezed out of a tube. It's flows. It's in fact, there's lots of other cells that you do not see.
Yeah, it's just a simplified vision of what's actually happening. Yeah.
And the GFP allows you to see some cells and not other cells, so you can see the cells that you do see, you see more detail in them.
And there's other things going on elsewhere that might be the part that's not stained with the GFP that is still doing something interesting. I remember having a conversation with. Sorry.
The branching of genesis is not only genetic, but I think there's a lot of physicists who are studying the interface, surface interface splitting that contributes to the branching of, say, like the trees, the glaciers, the cracking in the ground and that sort of thing. So they're applying the algorithm on the biological system like branching morphogenesis and see how much of the physical forces of the different surfaces are applied in that system.
Yeah.
So there's another question. Sorry, like there are a couple of questions there, Shankar, if you want to pick up.
So there's a question here about which forms. So I guess this is possibly interesting across different sorts of species. So I don't know if you know about. Obviously, you're looking mainly at mammals, but people are interested in birds and possibly even also insects. So the question here is which part forms first inside a hen's egg, or possibly even just a general bird's egg. And I'd like to extend that question to how about insects and how about mammals? What's the first bit that forms?
Sorry, I was looking at which part forms inside the egg. So to address that question, it depends on the organism. So one of the first things about mammals is that they have a placenta. So one of the first things that mammals do during embryonic development is to build the placenta. That's as opposed to say to a frog, fishes, etc., or even chicken, where they carry their own yolk with them. They have all the material they need. But that being said, even the chicken, which has its own yolk, has to create some sort of a interface to start to absorb the nutrients from the yolk so that the embryo can grow. So in terms of what comes first, in most cases, it'll be not things of the, it'll be this, you'll be setting aside structures and tissues that can utilize nutrition, either in the form of yolk or from the uterus, so that the fetus can grow. So you're essentially building the placenta. And then you have the body starting to grow, what forms the actual fetus. Within the fetus, I'd argue, I think arguably it's either the blood, you'd say it's either the blood or the heart. Different people will perhaps come down on the side of one or the other, but it is essentially the blood or the heart, which would be the first tissues or structures to form. And the reason for that, again, is because you need your nutrition to be spread or to be transported around the body. So the blood is what, your circulatory system is what transports nutrition around your body. And the heart is what pumps the blood around the body. So it makes sense that they are the first structures to form. And then after that, the brain is a close second. The central nervous system, the forebrain, is a close second in terms of what starts to form. Obviously, of course, the process by which it forms is a long process of maturation. So the embryo, it isn't as though you go from nothing to a fully formed brain, or indeed from nothing to a fully formed heart. You have cells that will give rise to just nervous tissue, and those will then start to form neurons, which have to mature. They need to be organized before you can start, in any sense, starting to have any sort of a nervous function, which comes much later.
Okay, I like that as an adaptation to which came first, the chicken or the egg. Actually, what we should be asking is which came first, the blood or the heart.
But it's certainly not the brain.
But certainly not the brain. The brain is an afterthought.
Ooh. I'm sure the pun was entirely intended.
Don't forget to tip your waitresses. The bad puns are coming. So there's another question here, which is in the same space, I think. Which do you think came? Oh no, hang on. Which do you think a zebra has? Is it black stripes or white stripes? I mean, this obviously is not your research area, but
no, it isn't. I'm sorry. I'm thinking to see whether there could be an answer for that, and I think there might be, in the sense that of course, it gets a little bit philosophical, but arguably you need to form pigment. The black stripes are a result of actual active formation of a pigment. So if you don't form the pigment, you will get white. So you could, from that basis, argue that you have black stripes on a white background, and that white is, in a sense, the default skin color of a zebra, and that on top of that you have the stripes.
I think we're both doing a bit of that, really, aren't we? With our sort of, there's a little bit of white coming through with both of our beards, and it's sort of the default color might be. I think that's a really interesting way of looking at these kind of slightly fun questions, but it is a further example of frivolity and humour. Life is all about sort of these interesting, curious, quirky questions. So thank you for answering those. Thanks for a really interesting presentation to just explore your work. I'm just going to offer a very brief plug to everybody in the room. Shankar and Tomoko worked on this really rather lovely project that we created and released a few weeks ago called Digital Bodies Convergent Pulses, that I'm going to put in the chat box. It's an artistic rendition of how, well, it's sort of how a heart starts beating. So I'm going to just put it in here. And it's a 5-minute video, and there's a sort of half-an-hour Q&A session that Shankar and Tomoko were part of. And so if you want a little bit of light entertainment, this is a 5-minute video that is really rather beautiful with a soundtrack, a funky soundtrack. I would highly recommend that you watch this Digital Body Convergent Pulses. Thanks very much for coming. Thanks you two once again for a really, really fun event. And we'll look forward to seeing more of you in the audience at more festival events in the future. Thanks so much. Cheerio.
Thank you, Dave. Thank you, everyone.
Thank you, everyone.